Oogenesis is the process by which the female gametes, or ova, are created. The female gamete is called an ovum. Sometimes people will refer to female gametes as eggs, but the term egg can include more than one stage of development, and the definition of an egg also changes depending on the type of organism. For example, the entire prenatal development of birds occurs inside an egg, but in placental mammals, after the egg is fertilized and starts dividing, nobody calls it an egg anymore. So, we're going to use the more precise term for a female gamete, which is ovum, or ova for the plural form. Now you may recall, each ovum must be haploid and contain only one copy of each chromosome. You may also remember that in order to create haploid gametes, a cell must go through the process of meiosis, which involves replicating its genome and then dividing, not once, but twice. In theory, this would create 4 haploid gametes from a single diploid cell, like it does during spermatogenesis. However, this isn't the case for human female gametes. Let's go through the process of oogenesis to see how this works and how many haploid gametes are made from a diploid cell. The diploid germ cells that have the potential to develop into ova are called oogonia. In humans, all of a female's oogonia that she will make in her lifetime are created when she's still a fetus and hasn't even been born yet. In fact, about 1 or 2 months before a baby girl is born, most of her approximately 7 million oogonia die, and the remaining surviving oogonia enter meiosis I and become primary oocytes. These primary oocytes press the pause button on their development in prophase I, after they've replicated their genomes, but before they've made the first meiotic division. They stay arrested at this stage of development for over a decade, until the girl begins her first menstrual cycle. Then, for about the next 30 to 45 years, on a monthly basis, primary oocytes resume meiosis where they left off and complete the first meiotic division. When the primary oocyte does finally complete its first meiotic division, it divides the chromosomes evenly, just as you would expect. However, it does not divide its cytoplasm equally. Almost all of the cytoplasm remains in one of the 2 daughter cells, which becomes a secondary oocyte. The other daughter cell, which gets half of the chromosomes but very little cytoplasm, is called a polar body. The polar body is not a functional oocyte. Instead, it degenerates and dies. The formation of a polar body allows the primary oocyte to reduce its genome by half and conserve most of its cytoplasm in the secondary oocyte. The secondary oocyte still has 2 copies of each chromosome, so if it's going to become a fully functional ovum, it must undergo the second meiotic division. This division is also uneven, like the first one, with half of the chromosomes going to another very small degenerate polar body, and half of the chromosomes being retained by the ovum, along with almost all of the cytoplasm. In this way, the ovum achieves its haploid state while conserving as much cytoplasm as possible. In humans, oocyte development occurs in the ovaries. Each oocyte is surrounded by a number of follicle cells to create a follicle.